APOE4 since I started my postdoc in 2013. I now have my own lab studying APOE4 and its role in Alzheimer's disease. Um, I'm joined by a former collaborator, Eileen Ruth Torres, who's going to help me moderate a little bit. Um, so I gave one of these seminars once before. That one was more of a sort of broad overview of what happens in the brain during Alzheimer's and dementia. Um, this one, I really want to dive into APOE4 and what it's doing in the brain and why it increases Alzheimer's disease. Um, and hopefully you'll come away sort of understanding a lot more about APOE4 in general and what it does, and how to prevent that association between APOE4 and Alzheimer's disease. Um, If I can advance slides. All right, so my talk has two parts. The first one, understanding understanding why APOE4 increases the risk of Alzheimer's disease and what can be done to prevent it. Sorry, in the second part, what can be done to prevent it. Just getting my screen set up. So I'm going to start with sort of a broad overview, and I'm actually going to start with a very complicated, overly complicated slide. This is actually meant to be a poster, so no worries if you can't read it all. You're not meant to. But this is something that was put together by Alz Forum a number of years ago, highlighting a lot of the major research discoveries of the past 100+ years in Alzheimer's disease research. And I think it's instructive for a couple of reasons why you'll see. But I want to just highlight a few of them. The first one, maybe the most important, back in early 1900s, a woman named August Dieter in Germany was having memory problems and general cognitive problems, mood problems. Went to a hospital in Germany, was seen by a doctor named Alois Alzheimer, who continued to see Miss Dieter, and then eventually she passed away, and Dr. Alzheimer did the autopsy on her brain and saw for the first time and described for the first time, these are his original drawings, two pathologies in the brain, uh, some these plaque pathologies and these tangle pathologies, sort of how he described it. Um, Fast forward all the way to 1980, and we start to understand a little bit about what's going on here. For the first time, it's identified that the component of these plaques that are seen in every Alzheimer's brain is this peptide, part of a protein called amyloid beta. And the main component of these tangles that are seen in every Alzheimer's brain is this protein called tau. And then most of these other discoveries that are made for the next 30, 40 years are around in some way, for the most part, amyloid and tau. For example, in the early 2000s, we discovered, or it was discovered, created the first amyloid PET tracer, so the ability to actually visualize the amyloid in the brain. The first tau tracers, tau PET, the ability to visualize the tau in the brain. Um, and for the most part, these were um, the discoveries that were made that were all mostly around amyloid and tau. Um, and it would be easy to miss one of these discoveries here, way back in 1993, that this gene slash protein APOE4 is a major risk factor for the late onset form of Alzheimer's disease, um, and that its counterpart APOE2 is protective. Uh, that discovery was made by a man named Alan Roses, or in the lab of a man named Alan Roses at Duke University in 1993. Side note, if you want to understand a little bit about how brutal it can be to work in academia, Alan Roses is a good example. He about 5 years after making one of the most, the major discoveries of Alzheimer's disease research, he had to leave academia because his lab ran out of funding. Uh, But at that point in 1993, his lab put out two, I think three, but a couple of seminal research papers, um, research findings. The first one on the left here is basically showing in this larger cohort of individuals who developed Alzheimer's disease, and he did some genetics, which was much more complicated back then. Showed that people with one copy of this APOE4 variant, and even more so with two copies of this APOE4 variant, had a higher likelihood of getting Alzheimer's disease and developed Alzheimer's disease at an earlier age. And then he also discovered, published that he saw the APOE4 as a component in these amyloid plaques, which sort of gave some um, confidence to this discovery that APOE4 was doing something fundamental in this process. So, so what's going on? So we have these basically two observations that are somewhat separate, or at least on the surface seem somewhat separate. We have these pathologies that occur in the Alzheimer's brain, these amyloid plaques and neurofibrillary tangles. Here's a microscope image of it to go along with Dr. Alzheimer's drawings of it. There's also the other hallmark pathology of Alzheimer's disease is neurodegeneration. Um, and then we have these genes slash proteins, APOE2, APOE3, APOE4, that have this outsized role in the development of Alzheimer's disease. This is a more recent version of that original sort of cohort study. This is with thousands of people, um, pretty much as big as you can get in terms of the amount of participants that were included in this. And you can see really definitively, individuals with two copies of APOE4 develop Alzheimer's, unfortunately, on average, sooner. APOE3/4, APOE2/4, one copy. And then APOE2/2 carriers, interestingly, are almost completely protected from developing Alzheimer's disease. So what's going on? How do you sort of square this? How do you tie these two different things that are happening together? Um, so in order to explain that, I'm going to revive um, a analogy that I used during the first public seminar, which I think is helpful here. I'm going to go through it a little bit more quickly this time, just because I want to get to the APOE part. But the analogy that I used, because I think it's helpful for people that maybe aren't as familiar with biology, but I think at this point we're all very familiar with, geography and geopolitics. Um, so the analogy that I'm making is that the body, one human body, is like the planet Earth, like the world. And like the world, like the planet Earth has lots of different countries. The human body has lots of different organs. For example, the United States is an important country. The brain is an important organ. I'm not saying those are exactly the same, but that's the analogy I'm using. Um, and then, you have different regions of the United States that are known for different things or important for different things: agriculture, technology, industry, oil. Same thing in the brain. You have different lobes that are responsible for uh, different functions, essentially: frontal lobe, temporal lobe, occipital lobe, where vision goes on. And then if you zoom in on one of those lobes, like the temporal lobe, you get to a smaller regions. There's lots of smaller regions in the brains. For example, the hippocampus. Just like if you zoom in on the United States, you'll get to smaller states, like Florida. And then if you continue zooming in into the hippocampus or any other region in the brain, you'll get lots and lots of cells. Neurons, microglia, astrocytes. In the same way that if you zoom in on a state, you'll get lots of cities, such as Miami and Florida. And then if we zoom in one more level, basically zooming in inside of a single neuron, you get lots and lots of these proteins. And the analogy that I'm making is basically that proteins are sort of the functional unit of the cell in the same way that people are the functional unit of a city. And by that, I mean, for the most part, human beings are doing vital, very important things in every city. There are human beings that are, Uber drivers transporting other people around. There are doctors and nurses who are, healing other people. There are garbage men and women disposing of waste. And really in the brain, proteins play pretty much the same functions. They're responsible for transporting other proteins, for repairing other proteins, for disposing of waste. And for the most part, proteins are doing very good, very important things. But every once in a while, you do get some proteins that sort of go bad. Um, in the same way that you get some people that sort of go bad. In the first seminar, I used this analogy of these 1920s gangsters, which was sort of a fun way to depict some bad guys. Um, so in our brain in Alzheimer's disease, the bad guys, the sort of proteins that go bad are amyloid beta, which is technically a part of a protein, it's a peptide, and tau. And in the same way that, one of these 1920s gangsters maybe isn't that big of a deal, but if they sort of start acting together, they, form a gang, they can wreak more havoc. Same thing, you have these amyloid beta uh, peptides that are very sticky and they stick together and form these plaques. You have these neurofibrillary tau proteins that stick together to form these tangles. And then there's one more sort of distinction that I'd like to make here, which is these aren't quite as detrimental to the brain as each other, the tau and the amyloid. So the amyloid you can sort of think of as a gang that's sort of spread out throughout the United States. It's causing problems, but at the end of the day, the United States is functional. It's doing okay. Same thing, you can have a brain full of these amyloid plaques. Many people do as they age. And for the most part, your brain can still function. You can have a brain full of these amyloid plaques and still be cognitively unimpaired. But then something happens, or sort of downstream slash parallel to the amyloid plaque buildup. You get this tau protein that starts to go bad in a specific region called the entorhinal cortex at first. The analogy that I make here, this is actually meant to be Al Capone. Essentially, what I'm saying is that tau is this more sort of virulent form of a bad protein, like Al Capone was. It accumulates within neurons and basically causes more damage than the amyloid does, and it actually ends up killing the neurons. And, it like a gang sort of moving through the United States, it sort of starts in one region and then eventually, goes from the entorhinal cortex into the hippocampus, and then throughout the temporal cortex and into the frontal cortex, and eventually sort of takes over the whole brain or at least the whole cortex. Um, and unfortunately, it's really this because it's sort of happening uh, within the neurons and killing off neurons. It's really this process that it follows the same regional process, the atrophy, the neurodegeneration that occurs, follows that tau process more than anything. And then along with that, unfortunately, comes, you know, cognitive decline, first mild cognitive impairment, then mild Alzheimer's, then moderate, then severe Alzheimer's. So that's sort of our general overview of Alzheimer's that I wanted to um, sort of tell you guys about. One more thing that I'm going to add on to this because I think it's important now that we're here, it's informative. So part of this process, this sort of downstream process from amyloid to tau, is basically these phosphate groups add on to the tau called hyperphosphorylation. Um, and that sort of helps to sort of the tau to sort of misfold and clump together. Um, so it turns out one of these phosphorylation sites, um, amino acid 217, um, is where is happening sort of definitively downstream of this amyloid process. And it's also showing up, it's sort of entering the blood, the tau P217 containing tau. So what that ends up being, and you guys have, a lot of you have heard of this, obviously, is this tau P217 blood biomarker, plasma biomarker. And what it ends up being, sort of because of this sequence of events, is a really good marker, and it's confusing because it has tau in its name, but it's actually a really good marker for amyloid. So essentially, people with uh, who would be positive for like amyloid PET tend to be positive for this tau P217. And then it's also on top of that predictive because it is sort of this intermediate step, predictive of future buildup of tau and neurodegeneration and cognitive decline. Okay, but what I really want to do and what I really want to get to is APOE. So, um, so you all know, every cell in your body inside your nucleus, you've got I think 22 chromosomes plus X and Y or X or Y um, that contain, that is our DNA that contain, the code for for our entire body. Um, and so on one of these chromosomes, chromosome 19, which has thousands of genes, one of those genes is the ApoE gene. And the ApoE gene gets, as with every gene, transcribed into or translated into RNA and then transcribed into protein to create the ApoE protein. So in preparation for this presentation, I was trying to think really hard what is the best analogy I could come up with for ApoE to go along with our 1920s gangsters. Um, I'll stay current. I won't go 1920s, but the best analogy that I came up with, and I think it's probably the right analogy, is a truck driver. So, we sort of think of, ApoE gets overlooked because it's not the pathology, it's not, but like a truck driver, which, you sort of don't think about, like if everything's going right, they're doing a very obviously important job, sort of bringing around materials and different things that are needed for buildings and everything else. But until there's sort of a supply chain issue or a strike or something, you don't really think about them day to day. And that's pretty much what ApoE is. So ApoE is basically the protein component of a lipoprotein particle. So everyone has heard of HDL and LDL cholesterol, obviously. Basically, HDL is high density lipoprotein, LDL is low density lipoprotein, and then cholesterol is the cholesterol component of that. Um, so these are basically these lipoproteins are basically these clumps of lipids, of phospholipids, of triglycerides, and of cholesterol that basically travel through our bloodstream, supply essentially the our cells with the lipids that it needs to basically function, well, to function, but also basically every cell in our body has this lipid bilayer. So basically, it's like a truck driver bringing around wood and concrete that's needed for the walls of our house, the rooms of our house. Uh, Basically, that's what these phospholipids are needed for, and basically, ApoE is sort of the protein part of that that's sort of bringing it around and loading it into the cells. One other thing that I want to add into this, just because I think it's useful, is there's something called lipid rafts, which is sort of a compact part of this lipid bilayer that contains a lot of cholesterol. Um, and you can think of that almost like the doors and the windows. It's where a lot of different sort of activities, comings and goings sort of happen, are in these lipid rafts, which are cholesterol rich, which we'll get to later. So I find that the majority of the conversation online when it comes to ApoE and cholesterol and all that tends to center around the body, sort of the arteries and veins and liver um, and that makes perfect sense. I think that's what we're all comfortable with at this point with biology is sort of what's happening in the periphery. But I think what gets overlooked in this conversation or not focused on enough in this conversation is what's happening in the brain. And the reason that's particularly important with ApoE is um, so in the body, you basically have uh, lots of different apolipoproteins that all do similar functions. Obviously, they have some variations, but ApoE is sort of just one of these many apolipoproteins that do this function of bringing around the the important lipids to the body or to cells in the body. In the brain, there's really only one that is doing most of the work. There's a couple others that are active, couple other lipoproteins, but really most of the work is being done by ApoE. So, it's almost like, the Teamsters or something who's got, they've got an exclusive contract, mostly exclusive contract on lipid transport in the brain. Um, which isn't to say that what's happening in the blood isn't important for the brain. Obviously, the brain gets oxygen and gets glucose and lots of important nutrients and things from the blood. So, if there is problems with the blood or plaque buildup in the blood that's causing atherosclerosis or strokes or different things, that is going to affect the brain. But there is this thing called the blood-brain barrier, which basically creates this very sort of tight regulation that essentially doesn't allow anything that is coming from the blood into the brain, or anything big, I should say. Small molecules can pass through, certain things can pass through, but certainly one of these large lipoproteins containing ApoE cannot pass into the brain from the blood, which means that the brain has to supply its own lipids, supply its own apolipoprotein, which is ApoE. That's done primarily by astrocytes, which is this sort of support cell in the brain, has lots and lots of important functions, including supplying lipids to neurons and other cell types. So it sort of loads up the truck right outside of the astrocyte with all these lipids, and then that gets shipped, with ApoE to the neurons. The neurons take them up, and then the lipids get distributed and the ApoE, leaves. And this is sort of a two-way street. The ApoE sort of taking lipids from the brain is also very important. Essentially, especially during like injury and stress, uh, lipids can get peroxidated, these sort of oxidative stress uh, ramifications that they need to be cleared out. So that's done by ApoE as well. Um, and then oligodendrocytes. So these are the cells that sort of create this myelin sheath around the axons of the neurons to sort of help the electrical signal travel farther. Um, so those myelin sheaths are basically mostly cholesterol. So that is needed. The ApoE needs to bring the cholesterol to the oligodendrocytes. And then microglia, which are the immune cells in your brain, which are sort of scanning around looking for anything that's not supposed to be there, um, microbes or dead neurons or even like synapses that need to be pruned. They sort of gobble them up, digest them, and then expel what they don't need, including a lot of lipids. So during this process, an activated microglia will upregulate ApoE in order to get rid of these lipids. So essentially, in this way, ApoE is sort of doing this job, throughout the brain when things are going right. So what about ApoE2, ApoE3, and ApoE4? Um, Essentially, every gene in our body has lots and lots of mutations, these single nucleotide polymorphisms that are, very important for evolution. For the most part, a mutation in a gene or a variant between one and another does absolutely nothing. Sometimes it has a small effect uh, on the uh, protein function. Um, in the case of ApoE, it tends to have some big effects. So basically, you've got these two mutations that there's lots of mutations in ApoE or potential mutations, but two that are sort of most important. This RS 7412, RS 429358, and they encode this one determines whether this 158 is a cysteine or an arginine amino acid. This one determines whether this cysteine 112, I believe, is a cysteine or an arginine, so ApoE3 or ApoE4. So on the surface, it's very small modifications. So basically just two amino acids that are different between ApoE2, ApoE3, ApoE4. But they end up having some big effects on the protein function. So in ApoE2, it affects the receptor binding region. So ApoE2 doesn't bind as well to receptors. And ApoE4, it sort of causes this hydrogen bond to happen that creates this more closed conformation of the protein. So I was trying to think what the analogy for these are, and I've created some other characters here. Um, so so basically, the way I see it is ApoE3 is basically your super competent truck driver, always on time, always doing its job well. APOE4, I hope this doesn't offend anyone, is sort of a little bit our like less competent cousin who got the job. They're doing their job. They want the paycheck. They're showing up, but they're not doing it nearly as well as APOE3, or not doing as well as APOE3. And then APOE2 is sort of also not as competent as APOE3, but sort of, not as competent in different ways than APOE4. So let's start with APOE4. Um, the two main things that happen with APOE4 as opposed to APOE3 is decreased stability and decreased lipid loading. So APOE4 is more likely to get degraded by the proteasome, other things. Um, and it's also less likely to have sort of a full load of lipids, added onto it, onto the lipoprotein. So it's sort of, it's our guy here is sort of calling into work sick a little too often and not quite packing his truck the way he's supposed to. And that, for the most part, again, he's doing his job, he's delivering the lipids, but he's not doing it as well as ApoE4. And then one other thing that's happening, this is a little bit more complicated, perhaps, but I think it's very instructive, is essentially when the ApoE4 or ApoE3 or ApoE2 gets to the neuron or any other cell, basically what happens is the ApoE, that part of the lipoprotein, binds to the receptor, number of different receptors for it, including LRP1. Um, and then that whole lipid complex gets taken up into the cell. And then in this sort of what's called the endosomal lysosomal system, I think specifically in late endosomes, the lipids get cleaved off, get hydrolyzed from the ApoE. The lipids get delivered to where they need to be, and then the ApoE leaves. So ApoE3 does this perfectly well. It enters the cell, it offloads its lipids, and it leaves exactly how it's supposed to. ApoE4, what's been shown is that it doesn't do this as well. And one of the things that happens is because of this closed conformation, it actually has some bad reactions in the low pH environment, and it can start to unfold and aggregate in the low pH early endosomes and late endosomes. And then it sort of ends up more often sort of stuck in the lysosomes with the lipids still attached. So, sort of think of it as our APOE4 guy here is sort of a little bit too social with the other APOE4s and not unloading the truck as much the way it's supposed to, ending up in a part of the site that it's not supposed to. And what this ends up causing is this increase, this buildup of cholesterol inside of neurons and other cell types. Okay, what about ApoE2? So ApoE2, you would think, considering individuals who don't have who have ApoE2 are protected from Alzheimer's, you would think that it was just really good at its job. That's actually not the case. Actually, ApoE2 is sort of it because it has um, this receptor sort of modification here. It doesn't bind very well to receptors, so it's almost like our ApoE2 guy has sort of forgotten its ID, and it's just not led into the construction site at all. So, usually this isn't a problem, and obviously people are fine with ApoE2, although individuals with two copies of ApoE2, so they don't have that redundancy of the ApoE3, they are more prone to something called hyperlipidemia, which is basically where lipids build up in their blood because the lipids are not being brought to the cells. But in the case of Alzheimer's disease, obviously this ends up being okay, partly because it ends up sort of not delivering something that ends up being a little bit toxic, which is this cholesterol. Also, the ApoE2 is sort of has a better ability for something called cholesterol efflux because it's sort of hanging out there. When there is sort of available lipoproteins, cholesterol can offload onto it. So ApoE2 sort of also helps clear cholesterol from neurons and other cells. Okay, so why does ApoE4 increase the risk of Alzheimer's disease? Um, to some degree, it's just that. It's this, ApoE is just really important for every cell type, and lipids are really important for every cell type. And ApoE2 and ApoE3 and ApoE4, does this ApoE deliver or lipid delivery a little bit different. For the most part, it's fine. For the most part, ApoE4 is getting the job done. You'd be hard pressed to find many differences in a, 30, 40-year-old ApoE4 carrier versus ApoE3 carrier versus ApoE2 carrier. For the most part, it's getting the job done. However, with aging, especially, problems can start to magnify. And then when you add in things like obesity and diabetes and stroke and TBI and infections and all these other things that have been shown to sort of increase the risk of Alzheimer's disease, especially in ApoE4 carriers, some of these problems start to multiply, um, especially because, if if a neuron's injured, it needs more cholesterol for synaptogenesis and different things that maybe ApoE4 isn't doing quite as well. And then there's also potentially, or pretty definitively, a more direct route, I think, which is, for example, this cholesterol. I was talking before about lipid rafts, which are these cholesterol-rich regions in the lipid bilayer. Um, One of the things among many that happens in a lipid raft is the production of A beta. So APP, uh, amyloid precursor protein, which can be mutated in familial Alzheimer's disease, but isn't mutated in sporadic late onset Alzheimer's disease. It does seemingly produce more A beta in ApoE4 carriers, partly because of this buildup of cholesterol in cells, and that amyloidogenic process being more active. So that is one thing that's been found. It's also been found that um, the ApoE4 can seed the plaques. Maybe that's why it's actually found in the plaques. Um, and it also ApoE can help clear or be involved with the clearance of A beta, which APOE4 does less well. So all that sort of adds up to this, fact that individuals on average who are APOE4 have more amyloid beta in their brains, um, which this is sort of depicted here in a amyloid PET scan. That's especially true for APOE4 carriers. Another way to visualize this is essentially this is showing sort of a big study that was looking at amyloid in the brains of people by their APOE genotype and just showing that starting in an earlier age, APOE4 carriers accumulate more plaque, more amyloid plaque in their brains. So when we sort of go back to our schematic here of sort of amyloid that then tips off this process of tau that then tips off the atrophy, it's really in the cognitive decline. It's really this amyloid part, this very early part that is most affected by APOE4. APOE4 can have subtle effects on tau, subtle effects on atrophy, but for the most part, on average, an individual with Alzheimer's disease who has APOE4 versus doesn't, they for the most part decline at a similar rate. It's just that they get Alzheimer's earlier, probably because the timeline gets sped up because of APOE4 and amyloid. All right. Hopefully I haven't lost anyone. I don't know who's still sticking around. Um, can't see. Uh, but hopefully everyone's still following me. I'm gonna now jump into for the rest of the talk what can be done to prevent this, because obviously that is, what anyone who carries APOE4 or has a loved one or whatever the case may be cares about. So I think about this, or I've come to think about this as I don't think there's a one size fits all or a specific, like, this is going to be the thing that stops all Alzheimer's disease from happening. If that happens, that'd be great. I think of it more as sort of like different strategies have to be considered at different stages. So this is our schematic. Um, so another way to sort of show this is one of these, it's called a Jack curve. Um, that basically, this is very common in Alzheimer's. You go to any conference, you'll see this a million times in one way or another. But basically, all this is showing is the same thing as here, that you have this increase in amyloid, this increase in tau, this increase in, and then this increase in brain atrophy, and then this increase in uh, cognitive decline that sort of happens in this sequential pattern. And it's only once you get to a point where you have lots of tau and the beginnings of brain atrophy that you get to MCI and eventually Alzheimer's disease. So I think about this a little bit similar. Oh, and sorry. So, and then APOE4 just sort of speeds up that process, brings us to the APOE, the amyloid stage more quickly. So I think about this a little bit in the same way I think about cardiovascular disease. I'm 46, about to be 47. I started going to the doctor and, getting my cholesterol and other things checked and had high cholesterol, got the calcium, whatever scoring. Anyways, we have this process that in some ways have become controversial itself because everything is controversial, but we go to the doctor and we get our measures for our heart health checked early because that's when we want to try to intervene. And if our cholesterol levels and other levels are high, maybe we try to intervene with different lifestyle modifications. Maybe that works, maybe that doesn't. If that doesn't, we have the option of going on a statin or another therapy, which is someone's choice. But the point is, you try to get at it early because once you do develop, real problems, real plaque buildup, a heart attack, something else, then you have to do a stent or a bypass surgery or something more drastic. So the time to sort of intervene ideally is earlier. And I think we're starting to get to that model with Alzheimer's disease. And part of the thing that's going to get us there that is starting to be appreciated is that we now do have a ability in the same way they have an ability to measure our cholesterol and other things. We have an ability to measure essentially our plaque buildup, our amyloid plaque buildup in our brain by measuring this tau P217 and sort of seeing where we're at in that process. And ideally, I think same thing, sort of like if we can get it down or prevent it with lifestyle modifications, that's great. I think we're learning about whether or not or to what degree we can do that. Um, and obviously, some people, same thing with cardiovascular disease, by the way, some people have a genetic predisposition that maybe makes that harder. Um, and I think eventually, if we're not able to do that, then that's where therapeutics will come in. So, so I showed this during the first seminar. This is the Lancet Standing Commission's sort of broad overview, really thorough look at what's called these modifiable risk factors. And basically what they found is that 45% of dementia cases could be reduced just by addressing modifiable risk factors. I'm not going to go through them one by one just because I don't really have time, but I'm just going to go through them somewhat quickly. I went through it a little more in detail before, but essentially they sort of break it up into early life, midlife, and late life. Um, Early life, having less education, unfortunately. Midlife, having hearing loss, having high LDL cholesterol, having depression, having traumatic brain injuries, physical inactivity, diabetes, hypertension, obesity, smoking, and excessive alcohol. And then late life, social isolation, air pollution, and vision loss. They've sort of come up with this, basically done a lot of combing of the literature and meta-analyses and stuff to say that these are definitively increasing your risk of dementia. So these and multiple others and supplements and anything else I'm in favor of that can potentially slow down that process. In terms of how APOE4 is how APOE4 carriers are affected, in particular, I think we need a lot more. This is one of the areas that I think we just need a lot more research on. But from what I've seen from the little that's been published on this, APOE4 carriers do seem to be pretty sensitive and pretty responsive to changes in these modifiable risk factors. So this is just a few examples from the literature showing that, in particular, APOE4 carriers, but not APOE4 non-carriers who have had lower levels of lower intake of saturated fatty acids, higher intake of polyunsaturated fatty acids, had a less steep decline in cognition. This was a study, I think, by the same group, looking at people who had high cholesterol, and it was really the ones who responded most in terms of reducing dementia incidence were the APOE4 carriers, the ones that responded most to statin use in this case. Physical activity, this study showed that APOE4 carriers in particular had higher amyloid beta levels if they were physically inactive, had less of what they called leisure time activity. And then this study, which was very recent, which I really liked, was looking at this large cohort of individuals over time. And basically, they assigned this Mediterranean diet score, they called their med score, that was basically sort of adding up how much, fish and nuts and grains and whatever else that was sort of part of a Mediterranean diet. And they assigned people a med score. and they basically said that people with higher med scores, so better diet, had a much lower dementia risk and much better cognition, and that the group that was most responsive to this change in diet or this difference in diet were APOE4 homozygotes. So people with two copies of APOE4. So just showing that it's APOE4 carriers are very responsive to lifestyle modifications to help slow down this risk of dementia. But, and people have different views on these different things. I think therapeutic discovery is hugely important for this. Um, so this is sort of an overview that I gave last time of successes. I say in quotation because really it's a dismal view. There were some early successes in Alzheimer's drugs, and then just decades of failures, clinical trial failures. And only recently did we have a couple that, for the first time ever, slowed the rate of cognitive decline in people with MCI and Alzheimer's disease. Um, that said, they didn't slow it very much. So, these and their side effects. So these have become controversial, but I think it's important to understand them for a couple of reasons, partly for what they do now and partly for what they might do in the future. Um, so these drugs, these two drugs, Leqembi and Kisunla, which are, FDA approved. They're essentially antibodies, synthetic antibodies that get injected into your bloodstream. And like any other antibody, they go after a target and sort of clear it out. Um, and so they just happen to be targeting these amyloid plaques in the brain really well, actually. So this was, 20 years of research to get, antibodies that targeted amyloid plaques this well. And they basically, over the course of 18 months, this is from the phase 3 clinical trial of Leqembi, clear out the amyloid plaques really, really well. What they don't do as well, unfortunately, is slow cognitive decline. So this is again from this phase 3 trial. They did meaningfully slow cognitive decline compared to placebo, so that is important. But people who took people with MCI or AD who took these drugs, unfortunately, still did decline through Alzheimer's. So, the thing I want to point out and really highlight here is this trial, both of these trials were done um, on individuals who had MCI or early AD. At this point, the brain is full of amyloid plaques and it's got lots of tau pathology. It's got brain atrophy. There's already some memory loss. Um, so unfortunately, at that point, and I think the trials proved it, getting rid of the amyloid, no matter how effective that may be, doesn't slow down the disease. I think the hope for people who already have MCI or AD really lies in the tau antibodies that are being tested now. Um, so that's our Al Capone character here. Um, so there's a number of they're all in, I think, phase 2 right now, phase 1 or phase 2. Um, a lot of big pharma companies have their own antibodies um, for tau. And that really is our biggest, best hope for slowing down Alzheimer's disease. And this, just to give a little bit of hope, this is phase 1 data from this. It's technically oligonucleotide, antisense oligonucleotide, not an antibody, but this is a drug by Biogen. And basically, what they showed just in a couple participants from their phase 1 safety trial was that these individuals actually had a decrease in the amount of tau in their brain. So that's something that could potentially actually slow down or stop Alzheimer's disease. Whereas I don't think the amyloid is going to do that, at least if given at this stage. And then there's also the possibility, also in clinical trials, that maybe once you do slow it down with a tau drug, maybe you can use stem cells to rejuvenate the neurons that have died, and reverse Alzheimer's in some way. I think that's the future at some point, but obviously we're not there yet, unfortunately. Um, but I do think there's still hope for prevention, and I actually think I'm these things are not popular, so this isn't a popular stance, but I'm still hopeful that these amyloid immunotherapies might be um, effective at prevention, which is basically meaning if you give it to people who have plaque buildup in their brain, amyloid plaque buildup in their brain, but have not had any cognitive decline, can you slow down or prevent, especially the tau? If the tau hasn't developed enough, um, maybe you can stop or prevent that process from happening. Um, this is these trials are underway now. The AHEAD 3, AHEAD 4, 5 for Leqembi, the Trailblazer ALZ 3 for Kisunla. It's been ongoing for I think over 6 years. We'll get our first results in 2027 and 2028. And then I think we figure out whether or not removing plaques early, which is I think when they would need to be removed, have an effect, can slow down this process or prevent this process, or as other people hypothesize, you know, maybe something else is driving this, and the plaques are just you know showing up as a marker. Personally, I think they're playing an active role, but I think these tests will tell us. One thing that I should note, so a lot of people who are familiar with this now know that there's these what's called amyloid-related imaging abnormalities. So these are basically edema or hemorrhages, but basically bleeds or different things in the brain that these immunotherapies can cause, which they usually don't cause people to drop out of the trials, but they are, obviously nobody wants that. Um, uh, and unfortunately, APOE4 carriers are more susceptible, especially APOE4 carriers. Um, the reason why I think it's too soon to say, oh, these drugs are bad for APOE4 carriers is we don't yet know whether they're going to have the same levels of ARIA or ARIA at all. They'll probably have some ARIA, but the risk of ARIA, same risk of ARIA when you treat earlier. And the reason why that there's a good reason to think they won't or they might not is one of the reasons this occurs, one of the reasons this ARIA phenomenon occurs, these side effects, is one of the things that happens with Alzheimer's disease, especially in APOE4 carriers, is you get something called CAA, cerebral amyloid angiopathy, where you get the amyloid actually building up around the blood vessels, um, and uh, essentially what's believed to be happening is that the antibodies are attacking this amyloid as it should, and then that's recruiting macrophages and different things that are damaging the blood vessels. So if we treat earlier, likely there's going to be much less of these, so maybe that happens less. There's also going to be less vascular damage in general in the brain. There's going to be overall less amyloid being cleared out. So I think we have to wait and see whether or not these side effects happen if given early in before the actual cognitive decline. All right, and then I'm going to finish off with a discussion about APOE4. Um, you know, can you target APOE4 as a therapeutic? Um, you know, Especially obviously exclusively, but in APOE4 carriers or target APOE in any individual. Um, and there's a number of approaches that are being pursued. There's gene therapy. There's you can increase the amount of lipidation of APOE4. You can disrupt potential binding between APOE4 and A beta. You can increase cholesterol influx. you can correct the structure of APOE4. So I'm just going to finish off by discussing two of these, just because I don't have time to discuss all of them, gene therapy and APOE4 structural correctors. Okay, so uh, essentially, this has been something that has been talked about for a long time, pursued for the last decade or two. But essentially, this idea of gene therapy where you modify someone's gene, someone who carries the APOE4 gene, can you, um, for one, turn APOE4 into APOE2? That's sort of the holy grail that people have been trying and talking about. Basically, converting the thymine to a cytosine, I think, on both of these um, nucleotide sites. Um, Unfortunately, uh, that ends up being very difficult for reasons that I don't fully understand because I'm not a geneticist in that way. Um, but it just ends up being very hard, I think, in general, and especially for ApoE. I think that the most I've heard about happening is 20% efficacy, like 20% of ApoE4 being converted to ApoE2. But in general, and they're still trying in other groups, I have a collaborator who's working on this, are trying to make this happen, but it's hard. So other approaches, one is the knockdown of ApoE4. This has sort of become more popular, this idea that maybe just knocking down ApoE4 levels by 50% might be a suitable therapeutic approach. And if you think back to that sort of cholesterol buildup in the ApoE4 neurons, you know, essentially knocking down ApoE4 would, in theory, have that happen less. Um, so maybe that would be a suitable approach. There's also the possibility of just expressing ApoE2. It's pretty easy. So it's easy to knock down something. In the case of CRISPR, it's easy to overexpress something in the case of these adenoviruses, AAV. Um, so maybe you don't mess with the ApoE4 gene at all. You just add in more ApoE2. So the company that's really leading the way, the biotech company that's leading the way on this is a company called Lexeo Therapeutics. Um, they actually have a drug that completed phase 1, phase 2 in I think 15 APOE4 carriers with MCI or mild AD, um, where they basically did the third approach, which is just overexpress APOE2 in the brain of APOE4 carriers. And they showed that they could at least stabilize A beta levels, and they did reduce CSF phospho-tau to some degree in some of the patients, some of the individuals. Um, and now what they're trying, or in the pipeline anyways, is basically this combined approach where they overexpress ApoE2 and partially knock down ApoE4. So it's sort of a roundabout way of doing this first one, this conversion from ApoE4 to ApoE2. And then there's a whole bunch of other, at least when I looked up which companies were doing this, I found a whole other list, a long list of other biotech companies that are trying either gene therapy approaches or antibodies to knock down ApoE4 or antisense oligonucleotides. Some version of these approaches are being tested. And then the last approach that I personally am fascinated by is this idea of an ApoE structural corrector, which is basically sort of taking this closed conformation of ApoE4 and turning it into more of an ApoE3-like structure by basically adding a drug that binds very tightly to this pocket. Um, and this is actually a approach that has been around for over a decade. A couple researchers at UCSF, Yidong Huang and Robert Maley, first proposed this and first showed that it could be done. They actually found some ApoE4 structural correctors. Those, for various reasons, and I don't want to speculate, never made it to clinical trials. But it's still an approach that a lot of people talk about. Interestingly, there's a company called Alzion, who a lot of people are probably familiar with at this point. They have a drug, valiltramipastate, ALZ 801, that is basically this prodrug of a compound homotaurine. The prodrug means it sort of enters your brain more easily and then gets converted into homotaurine. Ostensibly, what they say that their drug does, and I'm sure it does do this, is block A beta oligomer formation. And they completed a phase 3 trial, a very ambitious, very well-designed phase 3 trial on hundreds of APOE4 carriers, um, with MCI or early AD. Unfortunately, the ALZ 801 did not meet its sort of final ultimate endpoint, which is that it didn't slow cognitive decline in all of their patients or all of their participants, but in a subset, and specifically the individuals who only had mild cognitive decline, they actually saw significant, statistically significant slowing of cognitive decline. Um, so that was a big deal in itself. I don't know exactly um, what their regulation process, their processes with the regulators. But interestingly, fascinatingly, really, another group, I think unrelated to them, um, published during this time this observation that homotaurine actually can act as an APOE4 structural corrector. So it's possible that some of this positive effect that they saw was because of this drug acting as an APOE4 structural corrector. Um, and then just to give a little bit of a plug, I've actually got a collaboration with a regular collaborator of mine, Shahan Kazil, who made a number of compounds that have, we believe, has APOE4 structural corrector characteristics that we're currently looking for funding to try to test out and bring forward as well. So I think it's an approach for people who maybe are not inclined to get gene therapy, to hopefully be able to achieve some of that same biological effect with a small molecule. All right. That is everything that I'm going to present. So I will end the talk with a request for donations. Part of the reasons why I'm doing these public seminars is it's a really well, part of it just I do enjoy informing the public about some of the stuff we know in the science world. But also, it's a really challenging time in academia. Federal government is not being super helpful at the moment. So I'm sort of exploring different ways of raising money, and these public seminars are one of those ways. So if you're interested in donating, I'd appreciate it. All the research, all the money will go towards research in my lab. I also recognize that I keep giving these seminars and not showing any data from my lab and then asking for money for my lab, which isn't a great strategy. I think next seminar, I am planning to show some actual research from my lab. But if you want to donate, I'd be grateful. If not, no worries at all. And with that, I'll leave this up. I think Eileen added the link in the chat as well for the donations. But I will stop sharing, and I am very happy to take any questions that anyone may have.